That leads us into various different types of cloning. Now that I've got the genetic material, now that I know this is the specific segment I am wanting to figure out what in the world it does, now I can do a bunch of different types of cloning. So I can insert this genetic information and maybe get a copy of it and really try to figure out what it really does. And this is where we are going to take a whole hell of a lot of time to do some analysis. We have two basic types of cloning, and I have them both underlined. Stuff is underlined in here, guys, and it's underlined for a reason, or it's bolded for a reason. These are things that you're going to be asked about on the exam. I don't have it written up and done yet, but there are a number of great, fantastic multiple choice type questions here in this chapter. We have two types of cloning that we are going to talk about, and you should be able to maybe differentiate between the two. I don't know if it would make a good short answer question. Kind of sounds like it does, but I'm not done making the exam yet, so I don't really know how I'm going to ask it. But we have reproductive cloning and we have molecular cloning. Reproductive cloning is trying to get the whole damn organism. All right. Did it with Dolly. They've done it with a few other creatures since then. We'll talk about that after we talk about molecular cloning. Molecular cloning is where we just take a short stretch of that DNA, clone it, and try to figure out what it specifically does, and we can insert that into various different organisms. Doing this is a heck of a lot easier than doing reproductive cloning because it's on molecular cloning is such a small scale. Okay, molecular cloning is such a small scale. In molecular cloning, we can do easily in bacteria to introduce the genetic material that we want to figure out what it does. When it comes to doing a whole organism, there's a lot of moving parts. There's a lot of moving parts there. So it's much more difficult to do. Our ability has gotten a hell of a lot easier, better at it, not easier, but better at it. But still, there's a lot of moving parts there. So let's look at molecular cloning here. You want to create multiple copies of a gene, PCR. Okay. PCR to get us a bunch of different copies. Okay. And then we're going to put those in a bunch of different bacteria and put them into a bunch of different organisms and try to figure out what they do. So we're going to get a bunch of copies of it. We're going to try to get those genes to be expressed. So we're going to put them into something that we know, put them in a bacteria, knowing what the bacteria's genetic sequence is. We can put it in something to where we know this bacteria will produce this specific sequence, this specific genetic information. And then we can study those specific genes by what the stretch of genetic material, what it actually does, what it forms, what it creates, what it does. Okay. So again, we're going to use bacteria for this first. Easily manipulated, easily grown, doesn't cost a whole hell of a lot. So we're always going to do this in bacteria first. Okay. To get it into something, we need a plasmid. What's a plasmid? Plasmid is basically a vector. A vector is something that delivers the goods. Okay. Like for malaria, the vector is a mosquito. Okay. The mosquito carries it, doesn't cause them harm at all. But when they bite you, they inject the malaria, plasmodium, and then it gets inside of us, gets inside of red blood cells and causes us to have the issue. So a vector is just a plasmid here, okay? It's just again, what's the DNA of bacteria? Circular, okay? So we can get that in there fairly easy. Doesn't have to cross over too many membranes, right? And we can get inside and we can deliver this specific segment to the bacteria. The bacteria will then incorporate that information into their genome. So now I can give that bacteria to the host that I want to see what it does. Okay. As I just said, as that bacteria divides, again, it's going to incorporate that new genetic sequence into their own DNA. And now we're going to have some magic happen. All right. Let's see here, what do I want to say? You may see it listed in some sites as fDNA. And fDNA just stands for foreign DNA. It probably means something else to some other people, but for us, it's going to mean foreign DNA. We call it foreign DNA because we don't want to call it DNA anymore because it's been manipulated, right? It's been manipulated. I have inserted a segment of a gene that was not naturally in that bacterium before, and now it's incorporated within. So now this is referred to as foreign DNA. But this is how I'm going to get the genetic information in genetic material into something so we can produce it. Plasmids are what we use as scientists to be the vehicle to introduce genetic material to a different creature. But plasmids are already there to begin with. That's one thing I want to make clear. We can manipulate them hella easy. So that's why we use them. But they're there and they can contribute favorable traits to the organism. And the most common one with bacteria is going to be antibiotic resistance. Okay. So these things can change very easily. And so that allows them to get these adaptations, these crazy genetic sequences that mutate. And so that's going to give them this ability to be more antibiotic resistant. So again, like I keep saying, they're very easy to work with. Okay. They use them now to harvest bacteria to create insulin. So this utilization of this concept of plasmids is something that is done quite a bit on a very, 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 very large scale. These are pictures from your book. Okay. So here is this DNA segment that we got here. We use some restriction enzymes. Restriction enzymes are there to cut the DNA up. Okay. Okay. So we have this appearance right here. Okay. This is why we use restriction enzymes. Okay. Cuts it open. We can target a specific segment. And now it is ready for the plasmid to insert what I want into the genome. Okay. It creates these things called sticky ends. What do you think sticky ends are? Sticky, right? So now I have this little sticky end. All right. And I'm going to read the DNA. And this plasmid is going to come in here and read that and then insert the genetic material. And that's kind of what I'm talking about here. Okay. complementary base pair. And then we seal it back up. Okay, we anneal it. That's what annealing is. Open it up, attach things back in there, and then get it back together like we weren't even there. So I can take, here's that plasmid right there. Okay. I can use some restriction enzymes, open it up, get my DNA ligase, insert it in there. So now we have it in there. Okay. This is going to be incorporated into here. We're going to get this thing to divide hundreds and hundreds and hundreds of times. This plasmid is going to incorporate its genetic material into the host bacterium DNA. And whatever gene we've spliced into, this was the gene that we were looking to copy. It's the insulin gene. Okay. Insulin is what allows somebody who's diabetic to get utilization of sugar. Okay. So it gets sugar into cells. And you can see the end product here after I clone this bacterium hundreds and hundreds and hundreds of times. I can get this thing to produce a very large amount of what I'm doing. Now, this is with bacteria. We don't call it transgenic organism at this point, even though it really is. But that's not what we call this individual. It's just bacteria that we've manipulated for an industrial use. Okay. When it comes